Hello everyone, today we're going to be going over part B-1 of the August 2022 Living Environment Regents exam. The last video we covered part A1 or 1A, which was multiple choice questions 1 through 30. Today we're going to be covering questions 31 to 43. Starting with question number 31. Let me just enlarge this, which asks, based on the diagram, only an electron microscope can be used to view which of the following, right? So this diagram gives us the viewing range of your eye, a light microscope, and an electron microscope. And it's asking us only an electron microscope can be used to view what, right? So our answer is only going to be the section where it doesn't intersect, right? So it's going to be from this section that way, right? Why? Because if it falls in this section and forward, it can be viewed by both the electron microscope and the light microscope. We want to view something that can only be viewed using an electron microscope. Only that. Not something that can be viewed with the light and electron, only electron, right? So what falls under that category? Let's see. Well, straight off the bat, bacteria I can cross out, right? Because bacteria, if you look over here, if you look where it's labeled, bacteria fall under both the light microscope and the electron microscope. Can't be mitochondria. Remember, a mitochondria is an organelle, right? And right here it says that the organelles, right, their range is from here to here. So that falls under the light microscope and the electron microscope. That's wrong. Animal cells, here it says plant and animal cells, which definitely fall in the range of light and electron. And that leaves us with viruses as our right answer. As you can see, viruses, this range is only covered by the electron microscope, thus that is the only right answer. Moving on to question number 2, it asks, tells us that a scientist is developing a system to remove harmful bacteria from a contaminated water supply. In order to trap the bacteria and prevent them from going through the filter, she must make sure that the pores in the filter are no larger than what, right? So if you don't know, pores in a filter, this pretty much a filter, it's kind of like a net, right? And it catches debris, it catches bacteria, it catches particle. So think about this as one giant net, right? Except the holes in this net are very, 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 very tiny, right? These holes are so tiny that they don't allow rocks to go through. They don't allow bacteria or viruses to go through them. They only allow water to go through them, right? So what they mean by pores is this empty space that only the water is supposed to go through, right? And if these pores are supposed to block harmful bacteria, that means that they have to be smaller. They have to be smaller than the size of the bacteria itself. Otherwise, if the pore is bigger than the bacteria can just slip through with the water, right? So ideally, what we want to happen is we want to have these pores, and we want them to be we want them to be big enough so that water comes through, but just small enough so that when a bacteria comes along like this, it doesn't go through, it gets stuck, right? Because they're much smaller than the bacteria, right? So we're going to be, obviously our answer is going to be a range that is smaller than bacteria. So if we follow bacteria up here, we want our answer to be smaller than about 1.2 or 1.5 picometers, right? So let's look at our choices. Choice number 2 is the only one that's smaller than that, right? Because here it's saying 1 picometer. 1 picometer is here, right? And the bacteria, the smallest the bacteria can be is here, right? So the picometer is the smallest choice out of all of these, right? 100 picometers would let anything through, right? 10 picometers would definitely let it through. 1 nanometer, that would let everything here through, right? When you're reading this, remember that it's going from largest over here to smallest, right? And out of all these choices, this one is the only one that's smaller than the size of a bacteria. That's our right answer. Number 33 says that for native human populations in tropical areas, the intensity of ultraviolet UV rays from the sun is strong, and the skin color is generally dark. Melanin pigments found in people with darker skin color help block the effects of UV radiation on skin cells. In tropical areas, the best explanation for having an increased melanin in human cells is that it does what? right? So why? First off, let's just try to look at this by ourselves, right? Why do we have melanin, right? Because melanin uh, helps block the effects of UV radiation, right? So obviously, melanin has some survival advantage, right? It helps block out UV, and since you're blocking out the UV rays, you're not getting skin cancer. You're being healthy, right? So let's see this. In tropical areas, the best explanation for having increased melanin in human skill cells is that it what? Does it increase the occurrence of mutations? Wrong, right? It blocks out UV radiation. Since you're blocking out radiation, you cannot have a mutation, right? Because random mutations often come from radiation, right? And if you're blocking that solar radiation from hitting you, how can you mutate those cells, right? Number two, provides a survival advantage. That's literally what we just said. It provides harmful rays from hitting your skin and damaging your cell. That's all. That is why so many people have it. Think about natural selection. It's an advantageous trait, which means it's passed down from generation to generation, right? It doesn't act as a feedback mechanism, right? Because your skin color usually remains the same, right? It's not fluctuating by the day. It's not fluctuating by the intensity of the sunlight. And four, it just does not produce antibodies. Number 34 says smoking increases the risk of certain cancers of the mouth, esophagus, pancreas, kidneys, and uterus. This finding would be most reliable if it was based on what? Right? So we want reliable data, right? Reliable data, that means we want a large group, a large sample size, right? And one that accurately depicts that smoking increases the risk of these cancers, right? The best way to look through this is just look through the answer choices, right? So number one says data collected from patients in one cancer research hospital, right? I would say that this is not very reliable because it's just one research hospital. It's a single place. Your sample is reduced to only one facility. Think about it. Maybe that facility only has like 5 patients with cancer, or maybe that facility is in a very like heavily polluted area where just cancer is a more common trait, right? And think about it. If you're only getting your data, your results from that one thing, from 5 people, your sample size is small, your sample size is limited, and it can be affected by just regular circumstances, right? 2 says research done by scientists in many different countries. Okay, this is definitely something that I want to consider in the future. 3 says reading the information on cigarette cartons, right? I wouldn't say this is right? Because where's the case studies, right? We want to directly correlate that smoking increases the risk of certain cancers, right? We want to look at people who have been smoking and who have cancer. We don't want to use a source that someone else has wrote. We want to see this as a physical thing that's happening, right? So, and then 4 says cancer information published on social media sites. That's also going to be wrong. Number 1, it's on social media sites, right? Misinformation can always spread. And number 2, we want physical things. We want actual people with all these different cancers. We don't want just someone else's words, right? That leaves us with number 2 as our right answer. Why? Because research done by scientists in many different countries. Guess what that is? That's a large sample size, right? So you won't be limited to like 5 people in one hospital. All these different countries have different factors, right? So you're not just going to have all of your data come from one town that's really polluted and where cancer is really prevalent, right? So that's the most diverse group. And honestly, when it comes to finding the most reliable results, you want the most diverse group. Moving on to number 35, it asks which diagram below represents the direction that energy flows through an energy pyramid, right? The answer to this is 1. Of course, you should know this. Pretty much what happens is each level, you pretty much lose about 10% of the energy available, and you lose that energy to the sun, or to heat, right? So number 2 and number 3 is wrong because it doesn't really show the fact that with each level you're losing energy, right? If this was the case where energy flowed up and flowed down, this wouldn't really be a pyramid. This would be kind of a square, because obviously if energy flowed up, then this level would have the same amount of energy as that level, right? And this number 2 is wrong because this is flipped, right? If the energy is this, pretty much each level here signifies less energy. This has the most and this has the least, right? So if you have least amount of energy, how is that energy then being made into more, right? Remember, you cannot create energy, right? So if you're starting with this small amount of energy, everything below that should be even smaller, right? But that would be wrong even from the start, because you know that energy flows up, right? So that leaves us with number 3. Number 4, we just said that energy has to flow up. So that leaves us number 3 as the right answer, right? Remember, energy pyramid, some of it is lost to heat, right? About 10% of it is lost to the environment. So you need those arrows that face out. And the second thing you need to remember is that it flows up, right? It starts with the producers like plants and grasses, right? Then that grass is eaten by herbivores like cows, right? And then those cows lose some energy to the environment or to heat, right? Right, and then those cows, let's just say they get eaten by wolves, right? And then they have less energy, right? So just remember that energy is lost per level, right? And it flows upwards. Number 36 says an evolutionary tree is shown below. Which conclusion is correct based on the evolutionary tree? Right. So really good way to read these is anything that doesn't reach the present is extinct. Every node is some form of split or some form of evolution, right? So from here, obviously, it goes B. Species B was here, and it obviously species B separated into species O, right? And it's separated into another unknown species, which then separated into those different species, right? That's a quick crash course on how to read that, right? And it's asking which conclusion is correct based on the evolutionary tree. Number 1 says all the species have certain DNA sequences in common. This is correct, right? Literally, if we look at this, everything is related to B, right? Everything originally originates from B, right? Species B came here, then it split off, right? And because it split off, everything here in this section is related to it, right? Because it all came from the same node, right? If we trace this line, as long as we can trace the line or trace a line to a species, that means it's related to it, right? I'm tracing this line, I trace it there, right? I'm tracing this line from B, I can trace it to R, that means R is related. I can trace it all the way to S, that means S is related, and T, right? So this entire section is related, right? But then it also branches off here, right? Remember that this branch also comes from species B, right? I can trace this branch, right? I can get J, I can get K. If I trace this branch, I can get L, M, and N, right? So essentially what this means is that everything at some point came from B. And since they shared a common ancestor, since they all came from B, that means they all have to have certain DNA sequences in common from their common ancestor, right? Species S is the best adapted out of everything. You can't really tell that, right? Because if it was the best adapted, why did species Q also survive, you know? Species A is a common ancestor of species L and M. Sorry, a common ancestor of species L is M. How, right? Can I draw a line starting from M to L? Can I? Absolutely no, right? I have to go back down, right? In order to do that. And that's simply wrong. You cannot go back down. Oh. I don't know why it's not responding. But anyways, regardless, my point was you cannot, right? Sorry for that technical difficulty. You cannot move from M to L without moving down, right? So long as you are, the general rule is so long as you can trace it upwards and upwards only upwards, only moving upwards, and you can connect them as a common ancestor, right? So that's wrong. Species O and P are more closely related than P and Q. O and P, do you see the difference, right? Versus P and Q. Let's see. So O and P, right? That's one split and that's two splits. There's two splits separated, right? Meanwhile, P and Q are only one split separated. So there's only one node separating them. O and P, if we go back down, that's one node, that's two nodes. There's two nodes separating them, and the more nodes that there are between two species, the less related they are. Moving on to number 37, cute photos. Which two rows best Which two rows best support the information provided, right? So an Arctic fox has a gland in its brain that secretes a hormone that regulates the production of melanin, right? That's pigment. Always remember, melanin equals pigment. A pigment, we just said that accounts for brown fur, right? So they release a pigment, right? They release a chemical, right? And that chemical gives it brown fur, right? In the winter, the foxes secrete more of this hormone and their cells stop making melanin, right? So they appear white. The photos below illustrate two variations of fur color, right? So what happens in the winter, right? They have this hormone that regulates the production of melanin, right? So melanin equals brown fur. In the winter, they release a hormone, which is a protein, right? That stops producing melanin. It stops producing melanin, and because there's no melanin, they have white fur because there's no brown fur, right? Number 37 asked which two rows best support the information provided, right? So let's see what we just said, right? A, winter increased melanin, white fur. That's wrong, right? Because if there's increased melanin, we just said that melanin equals brown fur, right? Summer, increased melanin, brown fur. Sure, that's right. Winter, decreased melanin, white fur. That's true. And summer, decreased melanin, brown fur. That's wrong. Because the lower your melanin is, right? The whiter the fur will be, and the higher the melanin is, the browner the fur will be, right? So literally just take into account what they just said in that problem and apply it down here, right? Winter, it should be decreased melanin, so that's wrong, right? In the summer, it should be increased melanin, so that's also wrong. B and C are the only two choices that match the season with what happens, which you can definitely find by reading here, and it could also matches the outcome, right? So moving on to number 38, it asks which statement is the most likely explanation for the color differences in the fox fur at different times of the year, right? So number 1 says that mutations can be caused by changes in the number of biotic factors in the environments. That's wrong, right? Because as we just said, color differences come from the secretion of melanin, right? It comes from the temperature of the environment and the season. Season and temperature are abiotic, right? So that's wrong. Biotic are living factors. Temperature and time are not living, right? Number 2 says the expression of genes can be modified by the external environment. This is true, right? Literally, what we have been saying is when it's winter, right, their genes are expressed, the gene for melanin, or the amount of melanin production is lowered, right? So that is the external environment, aka the time of year, or the environment, which is a winter environment, which is altering an expression of a gene, aka melanin. Melanin is a gene, right? 3 says hereditary information is contained in genes located in the chromosome of the cell. This has nothing to do with hereditary because their fur changes, right? They're not. If it was hereditary, that means that it would have to do with the parent fox's, coat color. Number 4 says random changes in DNA can occur. That's mutation. We never talked about mutation. Guys, this is a super simple problem. All the answers can be found in this blurb right here. You just have to think about it, right? Number 39, and then the last question is 43. Okay, so number 39 says which statement describe? Oh, well, first let's read it, right? A new technique to attack flu virus antigens is being tested on mice. Normally, antibodies attack the head portion of the antigens found on the surface of the flu virus, right? So this is the antigen, right? It's the head, right? That's the antigen, right? Or this whole thing is an antigen, right? And that's just the head, right? So since the head protein mutates frequently, the antibodies do not provide protection for very long. This new technique to develop antibodies to attack the stem portion of the antigen. Since the stem regions do not mutate very often, the effectiveness of the vaccine should last longer. The technique is represented below, right? So let's just see what's happening here. So the traditional approach is that for the vaccines produce an antibody that bonds to the head, right, of the antigen. This whole thing is an antigen, right? It bonds to the head, and then it kills it, right? But over time, this head is very prone to mutation, right? So let's say in the next 3 months, this head mutates to have this, right? Now it's a polka dot head, right? And this same antibody that's specifically designed for a solid head can no longer bond, right? Because the polka dot head has a different structure, right? All because this mutated frequently, right? Now what happens here is that instead of attaching to the head, it attaches to the stem. And the stem does not mutate very frequently, right? So here, 3 months is passing. It's having a light gray head. It's having a black head. It's having a polka dot head, right? And all this time, the antibodies still work, right? Because the stem isn't changing, and because the antibody docks to the stem, which is always the same, right? So number 39 asks, which statement best describes an observation that would best support the continued study of using antibiotics produced by this technique against the flu? So, which one of these statements is an observation that says, "Hey, this is effective. Hey, this is good. We're doing something good. This is working," right? So, which one says that? Well, number one says a group of 50 mice with flu antibodies formed using the new technique were exposed to mutated forms of the flu. None of the mice became ill. Well, this supports it, right? Think about it. 50 mice, that's a large group, and they had flu antibodies formed using the new technique. All this means is that they had flu antibodies, right, that attack the stem. The new technique is using flu antibodies that attack the stem. They had stem attacking flu bodies, right? And they were exposed to mutated forms of the flu, which means that they were exposed to all of these different variations, the gray head, the black head, the polka dot head, right? And despite them having these different mutations, right, none of the mice became ill, which means that they managed to kill off the virus, which means that despite all these mutations, the antibodies successfully bonded to the stem and killed off the virus from the stem. This means that it's effective. This means that this new method actually works, right? This literally described what we found here, and it supports it, right? Number 2 says the use of these antibodies in mice stop mutations that occur in flu virus. This does not, well, sure, this is a good thing, right? But does it really support the purpose of the vaccine? No, the purpose of the vaccine is to kill it, right? This is simply just stopping mutations, right? Antibodies are supposed to kill the virus. This means that it's working for a different thing, so that's wrong. 3 says chemical tests showed that stem antibodies attached to the head of some heads of some flu viruses and destroyed them. This is wrong because this is saying that it's not doing its job. These antibodies are only supposed to go to the stem, right? So if some of them are going to the head, that means that it's not effective. That means that the antibodies clearly aren't working as intended, right? 4 says blood tests show that only stem antibodies attacking the stem of flu antigens can cause the flu in mice. Those attacking the head did not. This is completely wrong. This is the opposite. This is saying that those antibodies make them more sick, right? So number 1 is really the only choice that says that the antibodies work as intended, that they work despite mutations in the flu, right? Number 40 says scientists have discovered that pathogenic organisms and the chemicals they produce can cause foodborne illnesses. These illnesses harm the body as a result of interaction between the digestive and immune systems. Which statement most correctly describes how these two systems interact when an individual comes down with a foodborne illness? So what does this say? So when you eat something bad, let's say I ate a piece of rotten meat, right? There were organisms in this. There were pathogens, and when they entered my stomach, they released these chemicals. When my stomach started to digest them, they released these chemicals, right? Right, the chemicals that they produce, and the chemicals made my digestive system upset. Right, it made my stomach churn more, and it also made my immune system activate in my stomach, right? So, which correctly describes how the systems interact, right? Is what they're asking us. So, number 1, chemicals produced by pathogens enter the immune system through a cut in the skin. This is wrong off the bat, right? Because this is talking about foodborne illnesses. It's talking about, we just said how it is in the stomach, right? right? Because they harm the body interactions in the digestive and immune systems, right? This is talking about blood and the circulatory system. That's irrelevant to our problem. Number 2 says when specific chemicals produced by pathogens enter the digestive system in contaminated food, the ability of the immune system to fight foodborne illness is reduced. This can definitely be a right answer, right? Because it says that the chemicals are being produced in the digestive system from contaminated foods, and that causes an immune response. Number 3 says when food contaminated with pathogens are eaten, the immune system prevents the pathogens from entering the digestive system. This is wrong because this just told us that complications happen when the pathogens enter the digestive system and when they create chemicals in the digestive system. And number 4 says the digestive system breaks down the pathogens in the contaminated food so they are harmless. If this were the case, then the immune system would never have to step in. But as we see here, the immune system did step in, right? So if you read this carefully, number 2 is the only answer choice that matches that properly. Moving on to number 41, it says that a live plant was placed in a closed container in a lab. Sensors were set up to monitor the levels of oxygen in the container over several hours. Right? And this asks, at which hour were the lights turned on in the lab? Guys, they're giving us lights, they're giving us oxygen, right? We should be remembering one crucial formula, and that's photosynthesis. What does that do? It takes light, right? It takes water, it takes CO2, and it converts it into glucose, and it converts it into O2, oxygen, and also converts it into ATP or whatnot, right? But mainly what we're focusing on is the fact that it's that of the O2, right? So what's happening is we take light, water, and CO2 and we turn it into the oxygen, right? Here, it's showing us the production of oxygen, right? And in order to make oxygen, we need light and water, right? So at which hour were the lights turned on? Well, if you think about it, in order to make the most oxygen, we need the most lights. So obviously the lights are going to be on in the lab wherever there's the biggest production or the biggest concentration of oxygen, right? So let's see where that hour is, right? Is it at hour 8, right? Well, think about it. At hour 8, the concentration of oxygen is already pretty high, right? This means we're really just looking for this spike. It seems as if this spike is really when the lights started turning on because it started producing it, right? So the real answer is 2 to this problem. Why? Because here, oxygen concentration was decreasing, which means we weren't producing anything all until the second hour. When the second hour hit, all of a sudden, instead of losing oxygen, like we were doing here, instead of oxygen leaving the container, we started to produce oxygen. Oxygen concentration came up, and because the container is more concentrated with oxygen, that means we're actually producing more oxygen, right? And this rise means that photosynthesis is undergoing, right? Number 1 was wrong because we just defined this as the turning on point. Number 8, right? The lights were probably turned off here because once again the levels are decreasing, right? At 0, the lights had to be turned off, right? Because once again it's decreasing. And number 4, if we see here, this is the concentration of oxygen is starting to go down, right? So photosynthesis is starting to slow down, which either means that the container is fully concentrated, or it means that the lights were turned back off, right? That leaves us with number 2 as the only right answer. Number 42 says that during the 8 hours studied, the plant performed. Guess what? We were just talking about photosynthesis, guys. Right? Now, don't get confused here, right? Because there is a trick answer. A lot of people are going to choose photosynthesis only. That's wrong. Remember, photosynthesis goes hand in hand with, drumroll, cellular respiration. Because guess where this glucose is going to? This glucose is going right to the cellular respiration, right, in order to make ATP. So both photosynthesis and respiration are always going on at the same time in most plants, right? That leaves us with 43. It said that human cells have many molecules attached to their surfaces. Some of these molecules are involved in producing symptoms associated with allergies. Histamine is a chemical produced by some human cells. When histamine binds to the molecules on the cell surface that line the nose and throat, the cells will swell and leak fluid, causing the characteristic itching, sneezing, congestion associated with allergies. A model of this mechanism is represented below, right? So here's a throat cell, and what happens with this throat cell? It has many molecules attached to the surface, right? So this is one of those molecules, and what happens is histamine, right? Whenever we have like say pollen here, pollen in our cells, it enters our body like through our nose. Some cells are going to start producing histamines, right? And these histamines are going to travel all throughout your body, but especially where your respiratory system is, so especially where your nose is, where the most air is coming, right? And these histamines specifically bind to these sites on your throat cells, on your nose cells, and when they bind in, they trigger fluid release, right? So antihistamines are medications taken to block this reaction. Which antihistamine molecule represented below would be most effective? The answer is number 4, right? Because if we want to block antihistamine, we want to block this reaction. Guess what we're gonna have to do? The easiest way to block this reaction, of course, is to make sure that there's already something bound to these cells, right? Because guess what? If we have something that fits in here, and we have a histamine molecule, where's the histamine molecule gonna go, right? There's no space for it to go because all of these other places are taken up, right? So the histamine molecule can't bind to this, and because it cannot bind to this, it cannot release, the cell itself cannot release fluid. So the histamine's gonna have to go somewhere else, and it's gonna go to another cell, right? That either has these open, or if your antihistamine is good enough, it's also going to be blocked. So what's gonna end up happening is this histamine has nowhere to go because we just blocked it, right? A lot of you may be asking, what makes the circle so special? Why can't it be the triangle, the square, whatever this shape is, you know? It's because this is a receptor, right? And certain receptors only allow certain proteins, right? Or certain molecules to bind to them. And the shape matters. Shape always matters when you have these signaling molecules binding together. For example, let's just say that this was our signaling, this was our receptor, right? This receptor is specifically designed so that this chemical signal can bind to it, right? Not this. If this one binds to it, right, it cannot bind to it, right? You're going to be left with an awkward fit, and it's not going to activate, right? So when looking at this problem, we're choosing the shape that would most effectively fit into this little round little cubby. And the thing that would fit into this round cubby would be a circle. That's all, guys. That's part B2. We're not going over that today. This was all, this is a pretty short section, but a lot of reading. You have to pay attention to the reading that they give you, but you could really logic this out, right? So that's this, that's that. If you have any questions regarding this section, please let me know in the comments below. If you like this content and you want more, please like the video and subscribe. That being said, I hope you all learned something today, and I hope you have a nice night. Goodbye.